Very good afternoon to all the BIPC second year aspirants. So, here we are. Today we are going to discuss about genetics short answer type of questions. We are covering two chapters, that is principles of inheritance and molecular basis of inheritance. Before diving into the concept, I would like to give you a brief intro on the books that are being launched from NEET World. So, to get the complete free access of all the materials which are published or designed by the experts of NEET World, you just have to do few things in the social media where you will get the soft copies of all the 4 books. The very first thing is follow us in both Telegram and Instagram. The channel name is NEET World 25. The additional benefits that you will get by following these channels is that you will be freely accessed with the questions, like replica questions of NEET and MCET, and you will get the creative content, like on the toughest topics that you are struggling out there, you will get more creative shots and reels in Insta. So, if you follow in Telegram and Insta, it will be useful for your MCET and NEET as well. And then you see MedWorld app. This app you have to install from Play Store, and in chat box just say hi. All the 4 PDFs will be sent to you in person. To know more about the books or the institutes or the programs running out here, you can call us to the given number, or you can visit to our official website, which is www.neetworld.in. That's all with this intro about books that are being launched. Each book will have all the important type of questions with the complete blueprint of 60 on 60 marks for botany, zoology, physics and chemistry. And each book is having the model papers, guest papers and previous year papers as well. All the long answer type of questions are designed with flow charts so that you can easily analyze the concept. Next, if we see, yeah, so here we go with the short answer type of questions. First, that is principles of inheritance. Principles of inheritance and variation. The mostly asked question is explain law of dominance using monohybrid cross. So, when we dive into the concept in the actual sense, from the monohybrid cross, Mendel has proposed 2 laws. One is law of dominance and second is law of segregation, which are important, where we will get statement based questions in the NEET and MCET as well. So, learn this concept properly. This question, according to the IPE, we got multiple times, like in May 2024 and August 2022. These are 2 times where this question has already appeared. So, if we see how we have to go through the answer, first explain the proper definition of law of dominance. The law of dominance will state what? When 2 alleles of contrasting pair come together in F1 generation in the form of a hybrid, only one of them will express itself while the other remains hidden or masked. This is the actual definition of what? Law of dominance. So, if we take 2 alleles with contrasting traits, let us suppose capital T and small t for tallness. Capital T includes tallness and small t indicates what? Dwarfness. This will mask the effect of the recessive allele. And then you see the expressed allele is known as what? Dominant allele, and the masked allele is known as what? Recessive allele. Rieger Mendel has demonstrated this law using what? Monohybrid cross. Using pea plants, what is the model organism here? Pisum sativum, differing just in 1 pair of contrasting traits. So, in this diagram or in this chart, in this schematic presentation, you have to give out first parental generation. First, you have to present out parental generation. Parental generation, you will see pure lines of tallness and pure lines of dwarfness, and extract the gametes from the parental generation, capital T, small t, and next we will get a hybrid in F1 generation. All the offsprings of F1 generation will show the dominant phenotype only, and next selfing of this will give you F2 generation. Okay, selfing of this will give you F2 generation, and then if you see the phenotypic and genotypic ratio, phenotypic ratio will be 3:1. Okay, phenotypic ratio is 3:1. Genotypic ratio is 1:2:1 is the genotypic ratio and phenotypic ratio of F2 generation. If it will be very impressive if you present this in a checkerboard as well. Okay, so here we go from monohybrid cross, parental generation, highlight the gametes, and F1 generation, that is first filial generation. All the plants are tall in the F1 generation. Selfing of F1 will give you F2 generation. The only thing that you have to remember for this law of dominance answer or else any answer from genetics is present your answer in a systematic way. First the definition, what is meant by dominant and recessive alleles, then the schematic presentation, explain the parental generation, first filial generation, second filial generation in detail. This experiment will prove what? Characters which are controlled by pair of alleles, one allele is dominant and the other will be recessive. In heterozygous condition, only the dominant allele will express itself. Okay, recessive allele will be unexpressed or hidden. Next we see out, define and design a test cross. What is a test cross? Okay, test cross and back cross are important, not just for IPE but also for MCET and NEET. Test cross and back cross both are important. What is test cross? F1 hybrid, okay, F1 generation hybrid or a dominant phenotype is crossed with the recessive parent. Okay, dominant phenotype or F1 generation is crossed with the recessive parent, then it is called as what? Test cross. Test cross is the crossing of an F1 hybrid which is heterozygous individual with a homozygous recessive parent. Crossing this with a homozygous recessive parent which is of parental generation in order to determine the genotype of the hybrid. Why we are conducting test cross? You have to highlight this for sure. Why are we conducting the test cross? Okay, this helps us to find out whether the dominant phenotype is homozygous dominant or heterozygous dominant. Okay, F1 generation is whether homozygous dominant or heterozygous dominant. How we will get to know about this? Here we go with the explanation. The test cross is special type of back cross if the progeny shows both dominant and recessive phenotypes in equal proportion. If both dominant and recessive phenotypes are in the equal proportion, then the tested individual will be heterozygous. Tested individual will be what, heterozygous. If all the offsprings show only one dominant trait, all the offsprings after cross, if they are showing only one dominant trait, the tested individual is completely homozygous dominant. Okay, so first how you have to figure out this? Explain what is test cross and what is being identified from test cross. What is being identified, genotype is identified, after which what you will get to know? Whether the F1 generation is homozygous dominant or heterozygous dominant. This is what you will get to know. So if all the offspring show only dominant trait, the tested individual is homozygous dominant, if not it is heterozygous. Same the case whenever you are presenting out monohybrid cross, dihybrid cross or test cross. Start out from parental generation. Again I am telling you, first you have to give out the parental generation, then give out the gametes that are passed from parental generation, then F1 generation, after which test cross F1 generation and recessive parent, then neatly present it in a checkerboard. Okay, you will always get 1:1 ratio. The genotypic ratio will be 1:1. Okay, if it is 1:1, then it is heterozygous. So a test cross is a cross between F1 hybrid and a recessive parent. We have already dealt with it. This determines the genotype of the hybrid or F1 generation. It gives the phenotypic ratio which is 1:1 when the hybrid is heterozygous. Completely though it is dominant homozygous. So clear with law of dominance and test cross, no ma? Any doubt anywhere? Yeah, tell me if you are having any doubts. Yeah, and now we see, mention the advantages of selecting pea plant for the experiment by Mendel. Okay, what is the advantage of selecting pea plant or Pisum sativum as model organism? Here we go. This is a very important short answer type of question asked in multiple years, and the reason for choosing Pisum sativum for monohybrid cross or by Mendel is being asked in the form of statements in your MCET and NEET as well. So here you see, this question has appeared multiple times, like in May 2022, August 2022, March 2020, March 2019 and March 2017. This many multiple years the same question has appeared. So here we go with first, distinct contrasting characters. P plant will show clear variation. Clear variations are being observed, like tall or dwarf, round or wrinkled, yellow or green, cotyledons, etc., which helped in what? Studying the inheritance in a precise way. Second is annual plant. Pisum sativum is an annual plant which has short life cycle. This will complete the entire life cycle in few months. Okay, this will complete the life cycle in few months. This allows Mendel to observe several generations quickly and easy to cultivate. Okay, this is easy to cultivate, which grows easily in garden soil, and this needs little care or fertilizer due to nitrogen fixing root nodules. And then you see bisexual or hermaphrodite flowers. Okay, bisexual or hermaphrodite flowers, where it has both male reproductive structure and female reproductive structure. Each flower has both stamens and pistil, which will enable both self fertilization, and this is easy for cross fertilization as well. Next, large and conspicuous flowers. Okay, large and conspicuous flowers. The structure of flowers made emasculation and pollination simple. Okay, this will make emasculation and pollination simple. Next, self-pollination ensures purity. Okay, where natural self-fertilization will maintain true breeding or pure lines. This will maintain what? True breeding or pure lines for the experimentation. Next, easy cross pollination. Okay, Mendel crossing is simple by removing what? Anthers. This process is addressed as emasculation. Okay, this process is addressed as emasculation. Fine, right? And then we see this produces many seeds. Each pod will give several seeds, providing large sample size for accurate statistical study. For accurate statistical study, this will produce what? Large sample size. So what are the benefits that benefits of choosing Pisum sativum as model organism? We have distinct contrasting characters. Second is annual plant. Third is this is easy to cultivate. Fourth, this is a bisexual flower. This will have bisexual flowers, large and conspicuous flowers. Self-pollination is present, easy cross-pollination, and then this produces many seeds. Okay, these are the advantages of choosing Pisum sativum as model organism. Now we see, explain incomplete dominance with example. What is incomplete dominance? Incomplete dominance, here we see, this is observed in March 2024 and 2023. 2022 you have a, this question has come 2017, 2018 and 2019, 2015 as well. This many years applied question, and mostly in MCET and NEET also you have multiple previous year questions on incomplete dominance, co-dominance, multiple allelism and pleiotropy. Okay. So these are the exceptions of Mendelian laws of inheritance. So these are asked multiple times. Here we see incomplete dominance is the condition in which neither allele is completely dominant or completely recessive. Okay, no allele is completely dominant or completely recessive. This results in an intermediate phenotype in the heterozygote. Okay, intermediary phenotype. So here you see, of course, we have to start from parental generation. Parental generation is red and white, which is homozygous pure lines. Gametes you see, next after parental generation it is gametes. F1 generation is heterozygous condition, right ma? heterozygous condition, and in F2 generation, if you see the phenotypic and genotypic ratio of F2 generation, it is 1:2:1. Okay, phenotypic ratio of F2 generation is 1:2:1. Clear, right? Clear till here? Any doubt anywhere, ma? Yeah. Next we see in snapdragon, that is Antirrhinum majus or 4 o'clock plant, that is Mirabilis jalapa. Cross between red white flowers will give you what? Pink flowers, as the dominant allele is not completely expressive. It is incompletely dominant, right, ma? So here you have to figure out the explanation properly from parental generation and then gametes, then F2 generation, then phenotypic genotypic ratio. What is the actual reason also you have to mention out clearly. This is how you will present the incomplete dominance, be it test cross, be it explaining law of dominance with monohybrid cross or incomplete dominance. You have to show a systematic representation from parental generation to the offsprings. Remember that carefully. And then define law of segregation and law of dominance. So this goes good if we compare, if we give out the answer comparatively. Okay, law of segregation, law of independent assortment. Law of segregation is proposed by Mendel after monohybrid cross, whereas law of independent assortment is proposed by Mendel from his dihybrid cross. So here we go with first law of segregation. The two alleles of a gene which will separate from each other during what? Gamete formation. This separates from each other during gamete formation, so that each gamete will receive only one allele of the pair. Next, law of independent assortment. Law of independent assortment. The segregation of one pair of alleles will occur independently of the segregation of another pair of alleles during the time of gamete formation. This is the crystal clear definition that we are giving out in the first phase of both law of segregation and law of independent assortment. And then you see, ma, law of segregation is also addressed as Mendel's first law or law of purity of gametes. Fine, right? This law of independent assortment is also known as Mendel's second law. Then in a heterozygous plant, this is heterozygous condition, right? Gametes which are formed are capital T and small t. Whereas in a dihybrid plant, where two characters are present, okay, yellow color and round seeds, okay, yellow color of the seeds and round and seed color and seed shape. These are the two characters that are present. So the gametes that are formed over here will be capital Y capital R, capital Y small y capital R and small y small r. These are the gametes which are formed. How to identify the types of gametes? Types of gametes, n number of times asked question in your MCET and NEET. The formula to identify types of gametes. Formula to identify types of gametes is 2 to the power of n, where n is number of heterozygotes. Here how many heterozygous conditions are there? 1 and 2. 2 heterozygotes are there, right? So 2 to the power of 2 is 4. 4 types of gametes are formed over here. Clear, right? Any doubt anywhere, ma? Yeah. Next you see, so these are the types of gametes that are represented. Next, law of segregation ensures that pure alleles are transmitted to the offspring without any blend or mix. Pure alleles are transmitted. Here if you see in law of independent assortment, this will ensure independent inheritance of different traits. Independent inheritance of different traits. Next, law of segregation. This deals with separation of alleles of a single gene. Okay, single gene separation of alleles is given in law of segregation. This deals with independent behavior of alleles of 2 or more genes. Next, law of segregation is seen in all sexually reproducing organisms. Law of independent assortment is seen in organisms showing dihybrid and polyhybrid inheritance. This will show dihybrid and polyhybrid inheritance. Clear, right? I hope this is clear with respect to law of segregation and law of independent assortment. If you want to add on, you can add on one more point for law of segregation, that law of segregation is universally accepted, whereas law of dominance will have some exceptions. Yeah, one more important question, that is to differentiate between the following: between dominant recessive, between homozygous heterozygous, between monohybrid and dihybrid. This is appeared in May 2018 and May 2015. Here we go. First, difference between dominant and recessive alleles. Okay, dominant allele and recessive allele. Dominant allele is the unmodified allele of a gene that expresses its trait even in the presence of contrasting allele. This is even in the presence of contrasting allele. This is expressed both in the homozygous condition and in the heterozygous condition. The dominant trait in the F1 generation, this is the dominant trait that appears in the F1 generation. In pea plant, if you see, tall is dominant. Recessive allele, if you see, the modified allele of a gene, this fails to express its trait in the presence of dominant allele, expressed only in the homozygous condition. This is expressed only in the homozygous condition. The recessive trait will remain hidden in the F1 generation. Okay, this is hidden in the F1 generation, but this reappears in the F2 generation as well. In pea plant, dwarf, that is small t, is recessive. This is all about dominant allele and recessive allele. Next, you see differences between homozygous condition and heterozygous condition. Here both the alleles are identical for a gene which is showing pure line. Here if you see in the heterozygous condition, the condition in which two alleles of a gene are different. This is represented as capital T or small t, whereas heterozygous will represent capital T small t. Okay, and this produces only one type of gamete. Heterozygous condition will produce two types of gametes. This is pure tall and pure dwarf. In heterozygous condition, this is hybrid tall in the pea plant. Next, you see out differences between monohybrid and dihybrid cross. Monohybrid will include only one pair of contrasting traits. Dihybrid include a cross involving two pairs of contrasting traits. Here organism is heterozygous at one locus. In dihybrid, this is heterozygous at two loci. Cross is in between capital T and small t. Fine, right? And here cross is in between two characters, that is seed color and seed shape. This produces two types of gametes, and here in dihybrid cross this produces four types of gametes. Clear till here? Any doubt anywhere, ma? Clearly you have to present out differences in systematic way in under 4 points. That will be impressive. Okay. Next, about codominance, about blood groups, and then a brief note on chromosomal mutations. Yeah, this ends the chapter. Explain codominance with example. Okay, codominance with example, which is appeared in March 2023 and August 2022. Here you see codominance is a phenomena which both alleles of a gene will express themselves equally in a heterozygote. Okay, neither allele is completely dominant nor recessive. What is the difference between codominance and incomplete dominance? In incomplete dominance, the dominant allele is not completely showing its expression. In codominance, both dominant allele and recessive allele are equally showing their expression. This is the primary difference between incomplete dominance and codominance. Here we go with first codominance. The heterozygote exhibits both parental traits simultaneously. Both the parental traits are simultaneously expressed. With blending, both traits will appear side by side. Okay, both the traits will appear side by side in the phenotype. Examples here you see, first ABO blood grouping in humans and seed coat pattern in lentils. First ABO blood grouping you see, individuals with IA-IB genotype, IA and IB genotype shows AB blood grouping, okay, where both A and B are antigens. A and B antigens, they are expressed on the surface of red blood cells. And here you see seed coat pattern in lentils. When spotted plant is crossed with dotted plant, F1 generation will show both spotted and dotted seed patterns. This is one more example of codominance. So, here in F2 generation you can see 1:2:1 ratio. This 1:2:1 ratio will be same for phenotype and genotype. Phenotype and genotypic ratio will be 1:2:1. And then we will go with key feature. The heterozygote differs from both homozygotes as it expresses the traits of both the alleles distinctly, that is equal dominance. Here you see how many are there, ma? Here we go with this. Codominance by taking what? ABO blood grouping. Mention how many phenotypes and genotypes are possible from ABO blood grouping. Find right. So, first 4 types of phenotypes are possible, that is blood group A, blood group B, AB and O blood group, okay. And based on the presence or absence, like if it is IA, that shows A blood group, IO shows again A blood group, IB alleles, combination of alleles is B blood group, IB and IO shows B blood group again, IA and IB, this shows AB blood grouping, IO shows O blood grouping, okay. So, this is of ABO blood grouping. Clear, right? Clear till here? Any doubt anywhere? Yeah. Next we will move on to brief note on chromosomal mutations and gene mutations. So, chromosomal mutations and gene mutations, we will have a comparative study. First, chromosomal mutations which has appeared in August 2022 and May 2022. Here we go with chromosomal mutations and gene mutations. Chromosomal mutation is nothing but what, ma? Sudden inheritance changes or sudden heritable changes that are observed. These are sudden heritable changes which will occur in the base sequence, DNA base sequence within the gene. Chromosomal mutations are discovered by Hugo de Vries, okay, in Oinothera lamarckiana, okay, and gene mutation is studied as a part of Hugo de Vries and mutational theory. This is studied under mutational theory. The effects of whole chromosome or large segments of chromosomes are seen in chromosome mutations, whereas in gene, individual genes or pairs of DNA effects are shown here. So, that is why these are known as what? Point mutations or frame shift mutations. Here, change in chromosome number or chromosome structure is observed. Chromosome number is denoted under what? Aneuploidy, polyploidy conditions. Change in chromosome structure is learnt under deletion, duplication, inversion and translocation. Next is examples under chromosomal mutation: Down syndrome, trisomy 21, Turner syndrome is seen under monosomy 10. Gene mutations, sickle cell anemia, this is due to substitution of a single base pair. Next, what are the effects of this chromosomal mutations? Many genes at once they are affected, okay. So, this shows the effect on the entire chromosomal segment. If you see gene mutations, the effects of only one specific gene or small DNA segment is seen. Next, this will cause large scale changes in the phenotype, chromosomal mutations, whereas point mutations or gene mutations, this will cause small scale genetic changes. This leads to major genetic disorders or chromosomal syndromes, and this this will produce genetic variations which are useful in evolution, and sometimes this causes hereditary diseases. So, with this we have finished out the complete short answer type of questions of principles of inheritance and variation. In our next class, we will figure out all the short answer type of questions from mol bio as well. So, to all the second year students, I would like to end up the session by giving you a strong note that be with the subject, learn the concept in the more detailed way, and be ready both for the subjective exam and objective exam. Okay, don't do this mistake key. You will mug up the answers for IPE and then you will start the objective preparation afresh. Don't do that. Okay, you have both objective and subjective preparation together by learning the strong concept, and presentation you can play with it. Okay, so this is how you have to go through the syllabus in this last moment. Fine, right? If you are having any doubts or still you want to know anything, you can drop a message in the chat box. I will be addressing all your doubts. Okay, thank you.